Introducing the SmartAmp, manufactured and sold by K.K.Danaform as a detection agent for the 2019 novel coronavirus. The novel coronavirus has spread throughout the world. Much research is being conducted to bring this outbreak under control. Amongst this research, Riken and Kanagawa Prefectural Public Health Lab have joined together to develop a testing technology that can quickly detect the novel coronavirus. What enables this rapid detection of the novel coronavirus is called the SmartAmp method. Developed by Riken, the SmartAmp method is a technology that amplifies and detects specific genes. It is one of the isothermal nucleic acid amplification methods in which amplification occurs at a constant temperature. Compared to the PCR method in which amplification occurs by raising and lowering temperatures, less time and energy consumption are needed for amplification. The SmartAmp method has made it possible to detect the novel coronavirus more easily and quickly with great accuracy. This is the SmartAmp method, a DNA amplification technology that will replace the PCR method. These are the main characteristics that distinguish it from the conventional PCR method. The first characteristic is detection time. Using the PCR method, the detection time for the virus is about 2 hours. However, with the SmartAmp method, the virus can be detected in about 40 minutes. The isothermal nucleic acid amplification method is what makes this speed possible. With the PCR method, amplification occurs through the elongation reaction due to repeated changes in temperatures. However, using the isothermal nucleic acid amplification method, amplification occurs at a constant temperature of 67 degrees Celsius. Without the need for temperature changes, the time required is reduced. The second characteristic is less energy consumption. The SmartAmp method does not require temperature changes and occurs at a constant temperature. Therefore, the required energy is less than the PCR method. Due to this, it is possible to downsize the necessary equipment. These are the basic principles of the SmartAmp method. We began by heating the genomic DNA to modify it into a single strand. At least 2 types of primers are required for the SmartAmp method. Here, the different colors represent where the primer hybridizes. We use an asymmetric primer with a characteristic 5' end. Let's focus on the genomic DNA where the SmartAmp method reaction first starts. The primer, called the turnback primer, binds to the target region of the genomic DNA. Enzymes act, causing a DNA elongation reaction. The 5' end of the turnback primer is designed to be complementary to a part of the synthesized sequence. Therefore, as shown here, the end has equilibrium between the folded back state and the straight state. Another turnback primer binds to the genomic DNA when it happens to be folded back. This is where the enzyme acts. The DNA synthesizing enzyme used in the SmartAmp method has strand displacement activity. In other words, the DNA elongation reaction occurs while unwinding the double-stranded DNA. Then, the previously synthesized strand is displaced and released. The other primer, the folding primer, binds to the released strand. The enzyme acts here in the same way as before, and the DNA elongation reaction occurs. The double strand formed here is also in equilibrium between the folded back state and the straight state. When it happens to be in a folded back state, the turn-back primer binds it. This type of intermediate is generated by the DNA synthesizing enzyme with strand displacement activity. Now, focus on the 3' end of the intermediates. Both intermediates are designed to allow self-priming by folding back at the 3' end. In the case of intermediate 2, the 3' end is a sequence derived from the folding primer. When this part is folded back and the elongation reaction proceeds from here, a strand with double length is synthesized. Since the 3' ends of the strands synthesized in this way can be further self-primed, once the intermediate is generated, very long strands are continuously synthesized. On the other hand, with intermediate 1, the 3' end has a sequence derived from the turn-back primer. This part is in equilibrium between a straight state and a folded back state. As in the case of intermediate 2, the enzyme acts in the folded back state, and the elongation reaction occurs here, and a strand of double length is synthesized. This reaction is repeated and results in a very long strand. In this way, DNA amplification occurs very efficiently and can be detected as a signal. In addition, as a way of visualizing amplified products of the SmartAmp method, we have adopted a method for fluorescent labeling of amplified products. In this case, a fluorescent labeling is desirable, in which the background is lowered by quenching when there is no amplification, and fluorescence is emitted only when the amplification occurs. We have developed ePrimer, a fluorescent probe that can visualize the presence or absence of amplification, and have established a highly sensitive rapid nucleic acid amplification system combined with the SmartAmp method. Because the SmartAmp method can make positive judgment by amplification detection, the data analysis is simple, and the SmartAmp method shows higher detection specificity than the PCR method, while showing the same high sensitivity as the PCR method. The minimum detection sensitivity of the new coronavirus using the SmartAmp method is 50 copies. It can detect the new coronavirus within 40 minutes. The SmartAmp method can distinguish between positives and false positives. Because it uses the nucleic acid sequence-specific fluorescent probe ePrimer, melting curve analysis after the SmartAmp reaction allows positive and false positive discrimination. Because the PCR method does not use probes that enable melting curve analysis, this analysis is impossible with the PCR method. This is the amplification curve of the SmartAmp reaction. When trying to make a judgment by the presence or absence of the fluorescent associated with the nucleic acid amplification reaction alone, 3 should be judged positive because amplification is observed even though the fluorescent intensity is low. However, when observing the melting curve after amplification, a peak in the melting curve was observed at a temperature clearly different from the positive control in 3. In rare cases, such a subtle amplification curve can appear. Because this is often due to accidental formation of primer dimers, melting curve analysis is used to distinguish between a primer dimer and a true positive. Here is the detective reagent for the SmartAmp method. We use reagents that are not easily affected by viral mutations. We will use the newly developed SmartAmp method for rapid detection of the novel coronavirus as a countermeasure against the novel coronavirus infection. We at K.K.DNAFORM will collaborate with Kanagawa Prefectural Institute of Industrial Science and Technology, KISTEC, and will promote the social implementation of Kanagawa's technological creation, the SmartAmp method.